My name is Dr. Brittany Carr. I am a biomedical scientist in the Department of Ophthalmology and Visual Sciences here at the University of Alberta. I'm an assistant professor and I have a research lab where we study frogs and inherited blindness. Neuronal degeneration is really important because a lot of diseases affect our body and our neurons. Parkinson's disease, Alzheimer's disease, and in the retina. Retinal degeneration is really important because it causes blindness. And most of forms of blindness have no treatment or cure, and so understanding why this blindness happens is the first step into developing future treatments or cures. I think how many things can lead to degeneration, and at the same time, sometimes how resilient cells can be, or how much damage has to occur for you to even notice degeneration is happening in the retina. That's probably one of the most surprising things. So, especially when it comes to your vision, you can lose over 80% of your light detecting cells in the eye before you even realize anything is going wrong. So I think that's always really interesting. We need to understand the processes going on in cells to figure out ways to treat all diseases effectively. Sometimes we do want cells to die, especially during development actually, where we're making many cells, and then these cells need to form circuits, and these circuits need to be pruned, and sometimes we do want excess cells to die. If cells don't die, you can lead to certain diseases like cancers. You can develop ocular cancers as well from over-proliferating cells, and that can actually cause the loss of the eye. So we study frogs because we're interested in the photoreceptors of the eye, and in frogs, they have the same retinal structure and retinal cells as humans do, but their photoreceptors are about 10 times bigger, and so they're easy to see and visualize under the microscope compared to humans or mice whose outer segments are significantly smaller. And they're really easy to genetically modify. We can do everything in a petri dish. We can make hundreds of animals in a single experiment, and tadpoles are kind of cute anyway. So we need to understand the processes going on in cells to figure out ways to treat all diseases effectively.